So, hello guys. Now, what class of foods does legumes like beans and peanuts belong to? If you said proteins, you are right. In this lesson, we'll learn more about proteins. Now, proteins are natural polymers, just like carbohydrates. Of course, a polymer is a large molecule made up of several small molecules called monomers. Now, proteins are composed of polypeptides or polypeptide chains, which are in turn composed of numerous amino acids linked together by bonds called peptide bonds. I believe you can still recall what amino acids are from our lesson on amines and amino acids. Peptides, which are the base compounds of proteins, are formed when amino acids react. Two amino acids react to form a dipeptide. Three amino acids form a tripeptide. And several amino acids will form a polypeptide. So, a protein is composed of at least one polypeptide chain. The protein myoglobin has only one polypeptide chain. This myoglobin helps transport oxygen around the muscle cells. Hemoglobin, a protein that transports oxygen in blood, is composed of four polypeptides, while the proteins in food are composed of numerous polypeptide chains. There's one thing about proteins that you should know. They have unique structures composed of many substructures. And it is for this reason that some proteins like keratin in our fingernails and hair serve different functions. Let's quickly review the structures of proteins. A protein has a primary structure. The primary structure of a protein is obtained from the unique arrangement of amino acids in its polypeptide chains. It's like the way the letters of the English alphabet are arranged. A comes before B, which comes before C. A protein's polypeptide chain has a unique arrangement of amino acids. For example, the polypeptide chain of human insulin has a unique sequence of 21 amino acids, starting with the amino acid glycine, which is linked to isoleucine, and then to valine, and so on. The secondary structure of a protein is the manner in which its chains are folded or shaped. The polypeptide chains of a protein are usually folded into an alpha helix structure or a beta sheet structure. The tertiary structure of a protein is the manner in which its entire chains, that is, its alpha helices and beta pleated sheets, are folded to yield the final three-dimensional shape of the protein. In the diagram here, you can see how the alpha helices, represented as straight ribbons, and the beta pleated sheets, represented by arrowhead ribbons, are linked to one another and folded to form the protein's beautiful three-dimensional structure. The structure of proteins is determined using several techniques, including X-ray crystallography and spectroscopy. In the laboratory, a protein can be hydrolyzed into the individual amino acids that make up the protein. And this is done by reacting the protein with concentrated hydrochloric acid and heating the mixture at 100 degrees Celsius for 24 hours. It can also be achieved using enzymes. Also, because the structure of a protein is very important to its functions, proteins lose their functions when their structures are altered. Proteins are said to be denatured when they lose their molecular structure. Denatured proteins become insoluble, coagulate, and precipitate out of the solution. We can observe this phenomenon when we boil an egg. The proteins in the egg white become denatured and clump to form a solid. Proteins can be denatured by high heat, changes in pH, or when they are treated with chemical agents like acids, alkalis, and solvents like ethanol. We've learned so much about proteins. You now know that proteins are polymers of amino acids. They have primary, secondary, and tertiary structures. They serve different functions and are denatured when they lose their molecular structures. I'll see you soon.